All right, so we're going to start. You need to be quiet. And we're going to, first I'm going to show you where to find some things that you're going to need soon. So we're going to go to the blog site, hopefully. All right, so one, if you go down here, and as of tonight, we will have 4, 5, 6, 7, 8, and 9. No, we'll have 4, 5, 6, 7, and 8 on videotape. If you missed any lecture or you want to review it, it'll be online. Where do you get it? Right there. Click on YouTube videos, and it will list Chapter 6, Chapter 5, and all of them, plus the introductory notes and the midterm review. And click on videos, and you'll get all 26. So, this is to go to the website. Go to the website. Below the picture, where it says midterm review, there's where it says click here, then click videos on the page you arrive. So you click there, then you click on videos, and you get all 26. And of course, you know, most of the time it takes about 3 little videos for the entire lecture. So, if you want to review introductory notes, you want to review Chapter 4, Chapter 5, Chapter 6, midterm review number 2, which if there's nothing else you look at midterm review number 2, video, because that tells you all the tricks of the word questions. And if you don't remember introductory notes, look at it lightly. So that is for the midterm exam. Now the next thing I'm going to show you is for the lecture after the midterm. A week from today, click on Comprehensive Final Exam. Okay, final exam content, and go down here and it says final exam notes, eukaryotic parasites. And so if you click on eukaryotic parasites, it starts off with all the different parasites, acellular and eukaryotic. And we're going to start with viroids, prions, viruses. And so you're going to go here and you're going to read this information. And the lecture next time is right there. Are you listening? Because I hear somebody else talking. Right there, where it says acellular or subcellular parasites. They're not made of cells, they're smaller than cells. So read about viroids, satellites, virusoids, prions. That's where you get the information. Is there anyone who doesn't understand where to get the information? Chapter 10 and 11, eukaryotic parasites. All right. So that's where we will start our lecture a week from today. And after we lecture the first day, we will cover prions, viroids, satellites, virusoids. That'll be it. Then the next time we meet, we'll go over general viruses, and then we'll pick out 5 viruses that we're going to go over in detail. Human papillomavirus, which causes cervical cancer and genital warts. We'll go over hepatitis A, B, C, D, E, F, E, G. There is no F. And we will go over HIV/AIDS. We'll also include a little bit on 2 other viruses that somehow slipped my mind right now that are, Southwestern Pulmonary Syndrome, which is unique to Southern California, and people get it from cleaning out their garages. And so we will go over those. So on the 22nd, you will bring your term paper in typed in perfect condition for grading. It will be beautiful and gorgeous, and you will hand it to me, and while I'm grading them, you will take the biweekly test over prions, viroids, virusoids, etc. And then by the time you leave that night, you will receive your paper back, and if it is typed in the right format, has the, you haven't cheated by making up data. By the way, remember, I've been doing the same micros for 23 years. So if you make it up, I know it. And I also know which test didn't work this term, and I didn't tell you. So if you say you got a positive, and I know nobody did, remember in science, you don't make up data. Whatever you got is right. Yeah?
I was going to say, why would anyone make up data? Wouldn't they just use what they got and then explain it?
Yeah, but you know what a lot of people do? It's like memorizing exams. They think they're supposed to get what the book says. So if they didn't get it, or they didn't do all their tests, they just make it up. And so I have to have it's a very serious thing. So you end up getting a 0 on the required part of the test, a required part of the course. That means you get a 9. So don't do that. We'll talk about it when I go over how to type the report. Everything about it. But once you get, oh my gosh, you're here? Are you crazy? Are you feeling all right?
Oh yeah, I'm okay.
Okay. Okay. Wow, that's great.
Oh,
Oh, it was the other. Oh, okay. Okay. Oh, I thought it was her. Oh, it's the other one on that side. Okay. You may have to do your lab stuff like at the. How about at the end of that table where there's going to be more light?
Okay, thank you.
And you can go read it out. She'll show you. All right. So anyway, if you have got all the information and it's typed correctly and nothing else, then I will put somewhere between 90 and 100 on your paper, and I will put comments like second paragraph of conclusion, fix this, spacing in reference page, something like, I'll put minor comments of things you need to correct. You take it home, you do the correction, you staple my comments to the top of the final draft and put it in the final exam packet. And then you will get the grade that you earned that day. A lot of people think that they can go home and redo the whole thing and get a higher grade. Nope. You get what you got. Unless it didn't meet minimum criteria. Then you have 12 hours to redo it and email it to me, and the highest you can get is 89.5. If you don't have it in on time, it's minus 25% for every day you're late. So it's extremely important, and we will be going over it and telling you how to do it next time. Anyone have any questions?
What's the average length of the papers?
Usually, well, it's very modular, which means that basically you look at what I have online and plug in your data and make up your own introduction, covering the same points, and that's it. So the abstract, of course, the title page is 1, abstract is 1, introduction usually is 2, methods table is 1, data table is 1, conclusion is 1, and references, so approximately less than 10 pages. And it's very easy to type, and we'll be going over it. Okay, so now we're going to torture you with chapter 7. And first. All right, so we're going to start on genetics, and everything in this chapter almost we have already gone over. So it's going to be a lot of a review, except for just a few little minor details. And Could I get you to come over here and let her work there because she'll be under the lights? Thank you. All right, so could you clip on that for me?
It doesn't work.
It doesn't work? They blew the bulb, or is it plugged into the wrong place?
No.
There's always something. All right, so anyway, this is genetics, and first of all, that's E. coli's genome right there. So it's kind of interesting. It's the organism we know the most about. And this is a review. What's the difference between prokaryotic DNA and eukaryotic DNA? And remember, you have a single circular chromosome. It's all exons, no introns. Every 3 letters, what's an exon mean? Nobody tell me what exon means. Every 3 letters of the genetic code stands for an amino acid or a stop signal. There are 3 stop signals. Now, any 4 bases, you have the ATGC, and 3 of those is equal to an amino acid, and you would think, since there's only 21 amino acids, you'd need 25 triplet codons. In reality, there are 64. And the reason for that is many different, the bacteria have been doing this protein synthesis for 3.8 billion years. So there's been a few errors, and there are multiple codons that stand for the same amino acid. And that means they can make mistakes, and it's not a big deal. So remember, exons is where there is a triplet code that represents the amino acids, and there are 3 stop signals. So there are 61 codes for the 21 essential amino acids and 3 stop signals. Remember that the chromosome is not wrapped in a protective histone protein. Eukaryotic rods are. When it replicates, it still replicates 5 to 3, but it does it in a circle, making Okazaki fragments with a leading and a lagging strand. Yes?
How many bases would there be? Is this for the amino acid? How many bases? Is it 3?
Yeah, same thing.
Same.
Yeah, every 3 codons is 1. All right, and remember that it's semi-conservative replication as it's always been. So these are the differences and similarities of eubacterial DNA replication. A single circular chromosome, no encircling histone protected, all exons, doesn't exist in a nucleus. Doesn't have a double membrane around it. When it copies itself, one strand copies itself discontinuously with Okazaki fragments, and one continuously. It still does all of the same function. Copies itself 5 to 3, template for making messenger RNA and ribosomal RNA. Transcription happens at the chromosome, not in the nucleus, because it's bacteria, they don't have one. And it's still, DNA is a double alpha helix. So the similarities are huge, very little difference. Here's some vocabulary for this chapter. Remember, any change in the organism's DNA is called a mutation. And anything that causes a mutation is a mutagen. And mutagens can be radiation, they can be chemicals, they can be viruses. And so any change in DNA is a mutation. And what causes that change is a mutagen. Now, the exact DNA that an organism has is its genotype. How the different genes work together to give you brown eyes or blue eyes, that's phenotype. So what you look like is phenotype. What the actual genes are, the actual DNA, is genotype. Remember that mutations, most of you think that mutations are horrible. They're always terrible, they're always deadly, but in reality, mutations are overwhelmingly harmless. Why are they harmless? One, we have multiple codons for the same amino acid. Two, mutations in The smallest protein we have is 120,000 triplet codons. And you think changing one of those is going to keep it from folding like it should? No. Change 20 of them, it's still not. So overwhelmingly, mutations are harmless unless they start at the initiator or the terminator, or they are frameshift, and we'll talk about that. We have limited repair enzymes. There are 3 different kinds of repair mechanisms in bacteria, 2 of which are really good, 1 of which is useless. And then remember that the definition of a carcinogen is anything that causes cancer, and every cause of cancer is caused by mutation. So, all carcinogens are mutagens, but not all mutagens are carcinogens. For instance, I got this vitiligo, this killing of the melanin producing in a cell, and it happened because I worked with phenol. In the early days in micro, we didn't have plastic gloves. We just poured phenol down our arms and worked. And phenol, we discovered later, is a carcinogen. And so it killed or had caused a change of melanin in one cell, and every time that cell reproduces, it passes on that. So I have these white areas. That's a mutation. It's not cancer. Of course, if I stand out in the sun, I have no protection, so I might get skin cancer, which, by the way, I did. So, anyway, remember that all carcinogens are mutagens, a change in DNA. Remember that cancer is 1,000 different diseases with the same name. What do they all have in common? They uncontrolled division. When cells pile up on top of each other, that is a tumor. If it seeds out to other areas, it's a malignancy. And normally cells, the moment they touch another cell, they stop reproducing. But if they've lost what we call contact inhibition, then that is cancer. All right. And then we're going to talk down here about the Ames test, and it uses a mutant. Some of you are mutants, sick, twisted mutants that should be destroyed. You saw those movies, right? About mutants. Okay. How many of you in here can't digest milk properly? 1 mutant, 2 mutants, 3 mutants. Come on, is there any more over here? 4, 5, boy, more than usual. 5 sick, twisted mutants. You realize you're a serious mutant, because we are called mammals because we produce milk, lactose, and yet you can't digest it. That's serious. Anyway, whenever an organism has lost a normal nutritional ability, it's called an oxytroph. So you may think I'm weird, but you're a mutant. Okay, so if you've lost a normal nutritional ability that all other organisms in your species have, then you are an oxytroph. And we're going to actually use an oxytroph to get rid of large animal testing.
And you can pass that on to your children, right?
Yes, it's genetic. So remember that if we're speaking genetics, we're always speaking codon, messenger RNA. You don't go something like AGT. You always speak in codon unless someone says, I'm going to give you an anticodon or an active triplet code. So whenever someone comes up to you and says AUA, remember, they're speaking codon, not anticodon. You know it's RNA because it's U. So remember, the standard language of genetics is codon. Several codons represent, look at this, all of these are the same amino acid. They're all leucine. And what's constant about this triplet codon for leucine? What's constant? U in the middle. So the only one that really matters is the U in the middle. You'll still get leucine. And this is because over 3.8 billion years, the cell made a few errors, and these have accumulated, and so there are multiple codons for the same amino acid. So if there is a point mutation, changing one letter, you'll still probably get the right amino acid. So it's kind of like a self-repair mechanism. Notice that eukaryotes and archaeobacteria all start with methionine. Bacteria, eubacteria, start with a different initiator. And these are the 3 stop signals. I can only remember that, this one, because it stands for University of Georgia, and they were a big enemy, UGA. So I know that's a stop signal. So where in a codon strand or messenger RNA strand is it serious if you get a point mutation? And the answer is initiator or terminator. If it's in the middle, it'll probably still give you the right amino acid. If it doesn't, it'll still fold because there's 120,000 of them for the smallest protein. So that's the concept hopefully you'll get about mutation. One letter wrong is called a point mutation. And UV light causes it, nucleotide replacement, all of those things are point mutations. Remember, overwhelmingly, they're harmless. The other kind, though, frameshift, is usually fatal for the cell. Now, what do I mean by frameshift? I mean that the code is like A, T, A, G, 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 and you knock out a T, and everything moves down, so everything after that is wrong. That's called frameshift. You're taking a picture frame and putting it over 3 letters. If you knock out this one, that means this is the 3 letters. So everything after it is wrong, and frameshift is caused mostly by radiation, chemicals, and viruses. So insertions and deletions, insertions is what viruses do. They insert themselves in an oncogene, and that is how viruses cause cancer, because you have these introns that are fetal growth genes. Just remember the lac operon? All right, so you have a regulator way up here producing repressor that keeps that fetal growth gene turned off because you're not a fetus anymore. You don't need rapid division. But if a virus inserts between the regulator and the promoter and pushes the regulator so far down the repressor can't find the operator, it de-represses the fetal gene, turning on a cancer gene. So this is called the insertion, viral insertion theory, and it has been proven to be true.
And you said that chemicals, and what else? Chemicals, viruses, and you mentioned a third one called
Radiation. Ionizing radiation. Now you don't have to know hardly anything about this. This is just classes of chemicals that cause mutation. But this one you do need to know. Somebody sent me the question on the Google Plus. Let me think of the question. Antiviral drugs are most like what type of enzymatic control? And it's competitive inhibition. Remember, antiviral drugs are fake RNA or DNA bases, and that's a base analog. So the first antivirus drugs that we had were we would feed the virus while it's copying itself fake thymine or fake guanine. It would make its progeny, but the progeny wouldn't be able to reproduce because it had a fake DNA or RNA base. So this was what the first antiviral drugs were. They were fake DNA or RNA bases. Acyclovir is a base analog for herpes. So whenever people get herpes outbreak soup and your doctor prescribes acyclovir, he's prescribing a fake base, a base analog. And the lesion that you have is exploding and releasing virus particles. You take acyclovir, those particles infect another cell, and when they assemble to explode another cell, they can't. And the lesion gets less and less and less because the new particles can't infect new cells. Okay, these are just chemicals that cause mutations. Let's get over here to radiation. remember that UV light has some characteristics you need to know about. One, it doesn't penetrate glass well. It only penetrates one cell thick. And it causes mutation by causing thymine dimers or pyrimidine dimers, where two thymine are next to each other. And instead of binding across and making messenger RNA, they make thymine double bond dimers. So that's how UV light causes damage. And UV light is one of the two mechanisms that we are absolutely sure results in cancer. It's called accumulation theory. Same thing for smoking. For you people that smoke, you're going to die of lung cancer if you live long enough. And if lung cancer killed you tomorrow after smoking a cigarette today, you wouldn't smoke. But because it takes 10 years of accumulation of damage to the amount that you exceed your repair enzyme's ability to repair, that's why people always say, well, I'm addicted to the nicotine, I'm going to stop in January. On January the 1st, I'm going to stop, and since it takes 10 years, just never get around to stop it. Remember, this is the only legalized, super addictive drug, nicotine. It is more addictive than heroin. It is harder to break a nicotine habit than it is a crystal meth or a heroin. So this is legal, and so it's very, very difficult. The new little, what do they call them, mechanical cigarettes, they still deliver the nicotine, but they don't deliver the carcinogen. So you maintain your addiction, but you do not get as much exposure to the carcinogenic agent in the tar, because it's not burning tobacco. It's just giving you nicotine. All right, so remember that ionizing rays, X-rays, gamma rays, nuclear rays, all cause deletions and insertions, and they are fatal. Wherever it happens, everything after that is screwed up. And so ionizing always causes frameshift, and it's always fatal to that cell. So every time you get an X-ray, you are killing some cells. And then remember, since genes can move from the chromosome to the plasmid and back, that is a change in DNA, so transposon or translocation is also a mutagenic process. So this is what's going to kill you if you're a smoker. 5-bromouracil. And don't fall for that all the marijuana stories. That marijuana is less dangerous and not addictive. It is actually more dangerous because there is no filter. So you're getting more 5-bromouracil. And it is addictive. If you've ever known anyone that smokes 2 or 3 joints a day, go sit down beside them. Go talk to them. Their brain is fried. So what produces 5-bromouracil? Any burning organic compound. So when you burn garbage, you're breathing in 5-bromouracil. When you charcoal a steak, you're breathing in 5-bromouracil. Remember, every time you burn organic matter, you're creating this that looks just like thymine and will be used in your body. Thank God we have the repair enzymes to fix it most of the time. But if you look at very polluted areas where there's a low cloud layer and there's lots of burning trash and things like that, like Peking, there's an extremely high lung cancer rate, even in people that don't smoke. Why? Any burning organic matter produces 5-bromouracil, which is a base analog for thymine. This is ACT, the very first drug for HIV/AIDS, and it was, again, a fake thymine. This is just point mutation. These are things you really don't need to know, except just know that they're frameshift. Here is the non-ionizing UV light. Remember, we're repeating. UV light doesn't penetrate but one cell. Here's a test question. Can you sterilize using UV light? Answer, air and surfaces only. because it won't affect eukaryotic parasites because they're more than one cell thick. But bacteria and viruses are one cell thick or less, so you can sterilize air and surfaces with UV light.
You can, right?
Can, yes.
And you said don't even worry about the acronym, just know that they're Frameshift.
Yes,
Don't worry
they're
about the
Frameshift.
details.
Right. All right, so remember that this is called a pyrimidine dimer. Don't be fooled by my putting up there. UV light causes purine dimers. Purine is a pyrimidine. Remember that these are cumulative. Anyone ever seen those little old ladies that play golf every day in Palm Springs? Oh my God, look at their skin. It looks like elephant hide. They block every day, but they're going out there and they're accumulating huge amounts of damage to their cells. And you can see it's horribly wrinkled and it's tough, and it's just very, they will get skin cancer because, remember, UV light is cumulative. It adds up. Ionizing radiation, X-rays, gamma rays, they cause those deletions, and they are usually fatal. So here's the repair mechanism. There's only a few things in this chapter that are important, and this is one of them. And that is, there are 3 ways in which bacteria repair damage to the DNA. The most important one is photolase or photoreactivation. Yeah, don't get your hair too big. Okay. It's called light repair, and I don't like that name because it doesn't tell you what it does. Photoreactivation does tell you though. So I like photoreactivation. It is one enzyme called photolase. And what happens is it's very kind of a conundrum because the UV light causes thymine dimers, but to activate the repair enzyme you need UV light. So UV light is required to activate the photolase enzyme. And what does it do? It just goes down the strand till it finds a thymine dimer double bond and pops it. So it's photoreactivation, using light to reactivate a messenger RNA strand. Okay, next is excision repair or dark repair. And this is several enzymes. And what they do is they come in at both ends and they find the damage, they back up to and cut it and throw it away and resynthesize a strand according to the inactive on the other side. Both of these are very useful. The last one is not worth a darn. It's called SOS repair. And it's when a bacteria has been irradiated with UV light for a long time in one area, so that one area of the DNA strand has hundreds of thousands of thymine dimers. And so what the microbe does is the repair enzyme comes on there and cuts out hundreds and glues the 2 good ends together, hoping that those genes weren't necessary for life. And remember, bacteria don't have any genes that aren't necessary, so this is kind of useless. It cuts out huge regions where there is thymine dimers and glues the 2 good ends together, hoping that the organism can survive with that huge amount of DNA deleted. And of course it can't. Based on this, I have an interesting question for you. Question 11. Which is more dangerous, DNA viruses or RNA viruses, and why?
DNA, because we already know that the mRNA is going to be
It's RNA. Because there's no repair mechanism for RNA, only DNA. So if you get a mutation with RNA, there's no way to fix it. But with DNA, you do have the repair mechanisms. Okay, so this is light repair or photoreactivation. One enzyme comes down here, pops it, and flips back to normal. This is excision or dark repair because it doesn't require light. Two or more enzymes work. They come, they find the area, they back up to, they cut the whole strand out, and they resynthesize according to the other side. So remember, one is multiple enzymes, one requires UV light, and there's only one enzyme. Okay, so now we're on theories of cancer. There are many theories of cancer. There are only two that have real supportive proof. One's called a viral insertion theory that we talked about, where a virus inserts its DNA in the middle of a fetal gene that is turned off, pushing the regulator that makes the repressor so far away the repressor cannot find its way back to the fetal gene operator, de-repressing it. Turning on a fetal gene when you're an adult means these growth factors that cause uncontrolled division.
Is the virus considered a
Mutagen?
vertical gene transfer?
No, it's horizontal.
It's horizontal still?
So this is the viral insertion theory, and the other one is smoking and what else was it? Smoking and UV light skin cancer, called the accumulation theory, where you put layers and layers of mutagens on and you eventually exceed the ability of your repair enzymes to make the corrections. And this is where you accumulate damage over time. So, I'm going to tell you a fairy tale, because I'm a fairy, okay? So here's the fairy tale. Once upon a time, a guy was running for re-election to Congress. I know, who would want to go there? They don't do anything but argue and fix something at the last possible minute after it does most of the damage. But how do you get elected? How do you get elected in a democratic country? There's 5 major ways. Have more money than God and buy the election. You know, you buy more advertising. Remember that woman that ran around against Brown? That owned eBay and spent more than anyone has ever spent in a California gubernatorial, started a year in advance. She almost beat him. Yeah, Meg, what's her name? Yeah. So, most of the time, if you outspend your opponent by 10 to 1, you'll win. Because they only hear your name. Number 2 mechanism is have a trick to hang your name on. Like people change their name to No More Taxes Don. Or they change their name to John F. Kennedy. And lots of people, I mean, think about this. When you got your last ballot, how many of the things you voted for, people you voted for, did you have any idea about? Because I only knew the top couple. All those judges, all those people on the committees and all that. All I do is this. I pick a woman if a woman's running. And I pick a minority if a minority is running. Because I figure they've already been screwed over enough, they understand. And I always vote against the incumbent. Because are they doing a good job? I don't think so. So that's how I fill out my ballot. And by the way, did you know if you skip it, your ballot is invalid? If you skip one, you say, I don't know any of these judges, so I'm not going to vote for any of them. It invalidates the whole ballot. California. So make sure, even if you don't know, they want you to vote on everything. So don't skip anything. Well, anyway, so number 1 was have more money than God. Number 2 is name recognition or some sort of trick to hang your name on. Number 3 is negative advertising, and it works, so it's used the most. Now, what do you do in negative advertising? Remember, we have a 2-party system. We say we allow other parties, but do you ever hear them? Do you ever see them in in what do they call those things when they joust on TV? Yeah, they're never in the debates. So nobody knows about them. They have very little money, so they can't get their candidates out. So there's basically 2 parties, and the 2 parties conspire to keep anyone else from running. And so, what if you have 2 bad candidates? That's what negative advertising does. I may have been caught with a little bit of thievery when I was a city councilman. I know I did use my credit card for my wife's vacation and her mink coat, but I paid all that back, and And look at my opponent. They found porn on his computer. He's a perv. Now, your choice is between a thief and a perv. Who are you going to vote for? That's what they do, isn't it? That's negative advertising. You point out something horrible about your opponent, and then you take some little light hit on yourself. Well, I did make this one mistake. And of course, that person looks horrible, and you look wonderful in comparison. So negative advertising is used so much because it works. Because Americans have a 30-second attention span. And if you can get and dig into the TV ad in 30 seconds, they'll remember it. And remember, they don't have to be telling you the truth. There is no truth in advertising law in the United States. So they can lie in these ads so long as you cannot prove that it caused monetary damage. If it caused monetary damage to a candidate, they can sue. Other than that, they can lie all they want. So you can't believe a thing that they're doing in their advertising. It's all slanted. So, that's the third way. The fourth way, and this one's rarely used, but it's the one they designed all elections around, is come up with a new idea that no one has ever come up with before that fascinates the public into thinking it could be done a different way. And the last time that happened was Mr. Clinton. He came up with workfare. Everyone was complaining too many people are on welfare. And the more kids they had, the more welfare they got. And everybody was all upset about it, Democrats and Republicans. And so he said, I have a new idea. My new idea is you can go on welfare because there needs to be a safety net. You can go for 5 years. And during those 5 years, you can go to college or you can be retrained. And we'll even take care of your kid. We'll have child care. But after 4 years, you'll get another dime. And everybody was going, damn, why didn't we think of this before? And it was one of the big reasons he won. So, anyway, the last method is be against something everyone's against. Terrorists or immigrants. Now, it's so funny. Why are people against immigrants? They pay taxes and they don't get anything for them. It doesn't make any sense. It's totally illogical. It's just because groups of people love to hate somebody and you got to pick somebody to hate. Because that way you can unify all behind each other because pick somebody. So anyway, this man from New Jersey wanted to get reelected, and he campaigned that he was against cancer. How many of you are for cancer? He got elected. I'm serious. His campaign was, if I'm elected, I will pass a bill in Congress that outlaws any substance made, sold, or manufactured in the United States that causes cancer in laboratory animals or humans. And everybody thought, wow, this is a great idea. Until they thought about it a little bit longer. You see, there are a lot of things that we like that cause cancer. Number one being alcohol. You see, you would have to ban alcohol. People don't like banning alcohol. Plus, alcohol's a prostitute. You do know that, right? Of that beautiful bottle of vodka that you paid $35 for, about 15 of that is taxes. City, local, federal taxes, which they then use to advertise, don't drink alcohol. Same thing with tobacco. What is it now, $7 a pack? It's more than that, I think, in some places. And what do? All that is taxes. It takes 25 cents to make a pack of cigarettes. The rest of it's taxes. Now, why do we tax it so much? One, we want to make it very expensive. Two, we had no health care. And so people that had no insurance went to the emergency room and sucked resources when they got lung cancer. So we wanted to be able to pay for these healthcare emergency rooms and people that are being served without insurance. And then that woman with a hole in her throat is commercial.
That is a horrible commercial.
I know. I hope it scares the hell out of you.
It does.
I'm not buying another one. You're freaking me out.
Well, you know, my dad
This is like
smoked
too much.
Camels and Pall Mall for 35 years.
That's the thing, you hear about so many people smoking for years. So.
Oh, he did. And he started coughing at night. And so he took all kinds of over-the-counter cough medicines, and it didn't work. And he went to his doctor, and the doctor gave him something stronger. And then he started coughing up green, and he couldn't stop the coughing. And so his doctor said, well, let's do a biopsy. Well, x-rays and then a biopsy. And they found precancerous cells. Now, he had stopped smoking 8 times before that, and we begged him to start again. He was a mean SOB when he was not smoking. He gained 15 pounds, he was nasty to be around. And it was just awful. My mom and the kids, we all begged him, please start smoking. You're awful. But when he got the precancer cell, that's when he quit. He never smoked again. But he told me for the rest of his life, whenever he smelled someone smoking, he would go over there and stand next to him and go, oh, God, give my left arm just for one. Never lost the craving. The only thing that actually really helps is that new, what is it?
E-cigarette?
No,
Oh, the one that turns blue.
no, I'm talking.
The electrical one?
Well,
Chantix?
Chantix. Chantix really works, but you have to be prepared for it because you will have vivid hallucinogenic dreams on it. I know, for free. But do the program, it really does work. It works. Never before have we had 80% of the people that try something be able to kick it.
Well, yeah, because they're
But-
like hallucinating.
Well, what is it that causes the hallucinations?
It's just the drug. They're using.
Is it ketamine they use in it?
I don't know. I don't know what it is. All I know is that you can smoke your regular program and you start it in very low amounts and then you add more of the Chantix and you lower your cigarette smoking and eventually.
After 1 week, you basically don't want to.
Yeah, you don't have to
I
break
I
it.
don't know from experience.
Well, I know from experience because I had an ex that successfully kicked it and he had been smoking for 20 years and he had tried everything before and it worked. But anyway, so tobacco is also, a lot of the taxes are used to advertise don't use it, which is prostitution, right? Yeah. Right. So the next thing they discovered is all cosmetics, we don't want to say, because men wear cosmetics too now, you know. So all cosmetics, and the cosmetic industry is billions and billions of dollars. And the cosmetic industry says all we do is smear it on the outside of them. So we shouldn't be subject to this law. And so they passed an exception that you can smear it on a rabbit, and if a rabbit doesn't croak or get cancer, you can sell it. So cosmetics that are not ingested are exempt. And of course, you know a lot of cosmetics is all hype, right? Lancome, Rainbow Seam, disappear. Seam? And you pay $50 a little thing for that? My mother still does. 88. Right? But for instance, lipstick is regulated because you can ingest it. In fact, when they first started this test, they found red dye, I think it's number 9 or something like that, that was a major color in red lipstick, had to be replaced because it wasn't discovered to be a carcinogen. So lipstick is regulated because you can ingest it, but the other things are not. And then, what do people without artificial sweeteners do? Because there weren't any for a long time. So they use saccharin. And we do know that saccharin causes cancer, is a carcinogen, but it's in such huge amounts and only in males. So if you ate 100,000 times the lifetime cumulative dose of saccharin, you could get a kidney cancer. And that's such extreme, and since there was nothing else, they allowed saccharin. Huh? Well, you know, that is NutraSweet. So,
Nutrasweet's not saccharin? No?
No.
The pink one? Sweet loss?
So what's the
the
equal?
yellow one is not saccharin, the pink one is half saccharin.
Half saccharin.
So what is equal?
Equal,
Equal's
I don't
good.
really know which one that
Equal, you can't tell.
is. Anyway, but saccharin was allowed, and then they discovered hydrocarbons cause cancer. Well, hydrocarbon is gasoline, kerosene, all of those. How are we going to ban gasoline? So what they did then, when they discovered that hydrocarbons and asbestos, which is in every ceiling tile and every floor tile, to prevent sound from vibrating around and because it retards fire, they said, okay, if removing that substance from the public would cause economic collapse, then all you have to do is put up signage. So if you read the gasoline pump, when you're pumping, it says, Cal OSHA, the federal government, had found that breathing gasoline fumes causes cancer, don't do it. And if you go into a garage, there's a sign on the floor. Our insurance company has said that the public is not allowed in this garage. Why? Because asbestos is found in brake lines and fan belts. Asbestos is a known carcinogen. Asbestos fibers have been found in this area. Don't breathe them. And I don't know if you remember, but when I was in school, all of our ceilings was this sort of spray-on stuff. In fact, we used to throw our pencils up there and stick them in the ceiling and watch the fibers come down. That was asbestos fiber. Yeah. So now it's, when you build a new building, you can't put asbestos in it, but what about all the buildings built between 1940 and 1970 when this went into effect?
Lead.
So, here's what they do. By the year 2020, they all have to have the asbestos either covered up or removed. If you remember, about 5 years ago, they shut down that Min building here to remove asbestos. They put a big plastic tin over it, and men in plastic suits went in there and dug out the asbestos. Remember, asbestos is a mineral. So what they do with it? Bury it, because that's where we got it from. Yeah.
The old USC building, the one that they don't use anymore, they said that the reason they don't is because of asbestos contamination. But they can't tear it down because of asbestos contamination either. Is it more dangerous if it's...
Yeah, the problem with tearing it down is it releases it into the general environment and everybody, and the wind carries it.
Yeah.
So for instance, at LACC, you know, it was once originally UCLA. That's the original site of UCLA is where LA City College is now. And we have a lot of the buildings from the late 40s and 50s there, and they're filled with asbestos. Well, there are two ways to get rid of it. You can dig it out, which costs about 100% of the cost of the building. In other words, you just come and wear a spacesuit and gather it all up, and put it in a plastic bag and carry it out. Or you can spray plastic over it. That's what they do there. They spray plastic over it. The problem is every time there's an earthquake, plastic breaks. And the fibers are released again. So it's not a perfect solution no matter what you do.
Dr. Hicks, so why is it that like if you go to any building, even if it's a new building, it says this build you're entering at your own risk?
Right.
Is it because of asbestos
Yes.
still, even if they're new buildings?
Well, not new buildings usually don't. There is banned now in new buildings. Where is this at?
Why is it down? So like they can open like a B of A branch and you walk in and there's like
Maybe they
But
use.
don't they put it up for everything now?
Yeah, so they get sued because,
Yeah.
yeah,
They
the
put it
carpet is
everywhere.
made of artificial fiber
Yeah.
and people get headaches and they sue. Yeah.
Like, don't drink this milk, you're gonna be
And a
cancer.
lot of parking garages too.
Well, that's because of the asbestos in the brake lighting and fan belts. So anyway, what did this amendment cause? It meant that every product in the United States had to be large animal tested. Because it says that anything that's known to cause cancer in laboratory animals or people. So my father was a polluter of the universe. His job was he designed herbicides. These are poison, plant poisons that when you plant a crop, you spray it at the same time. And the corn comes up, but no weeds come up. And so he had to prove that Treflan, which was his product that he developed, did not cause cancer. So when I was 9, I used to go with him to his lab, and I would play with 5,000 beagle puppies. They had them in runs, 20 in each run, just two or three acres of runs of beagle puppies. 1,000 of them got regular food, and the rest of them got different amounts of Treflan in their food every day for 5 years. So one day I went to visit, and there were no puppies. All the runs were empty. And I said, You know, because I used to name them, play with them, and teach them. Beagles are a wonderful breed, incredibly. Horrible bark. Oh. They are really, really great dogs. And I trained them, and I come home and smell like a dog. You know. And I went with him, and there were no puppies. And I said, Daddy, where do the puppies go? He said, 1, they're not puppies, they're 5 years old now. And 2, they're dead. We sacrificed them for the experiment. We had to autopsy them and compare those that got treplant with those that didn't. He tried to explain to me. I didn't speak to him for weeks. And so think about how expensive it is for every new product to go through this for 5 years. And then to look and evaluate all the autopsies and everything. So it was a very expensive process until 1980, when a man by the name of Ames designed something better that has almost eliminated large animal testing. And he took a salmonella that's histine negative. Now, this salmonella normally uses DNA to make this amino acid, which is needed for life. It has a point mutation, which means 1 letter in the DNA code is missing or out of order, and therefore it cannot make it. So unless you feed it in the diet, this salmonella will not live. Everybody get it? Normally it makes this amino acid, but this one is a mutant. If it needs something it makes and it's mutated, that means it's an oxytrope. And so here's what he did. He made up agar that has no histine in it. Then he plates the bacteria over it. Should they grow or not? No, they won't grow. All right? He drills a hole about the size of a pencil eraser in and puts any compound he wants to test. Sticks it in the incubator overnight, and they have found that any mutagen will cause it to revert to wild type, flip back. And so the next day, if he has no colonies, the substance is not mutagenic. If he has colonies only near the center, like a few around here, it's mutagenic at high concentration only. And if they're equally distributed, it means it's mutagenic at any concentration. Now, what's good about this? It's overnight, not 5 years. You can do it on billions of bacteria. Remember the rule of generalizability? The more individuals you do an experiment on, the better the experiment represents real life. It's cheap, $50. And you get rid of all mutagens, not just carcinogens. And no puppies have to die. That's my favorite part. No puppies. Now, if I was the tooth fairy, and I could wave my magic wand and get rid of carcinogens or mutagens, which one would you want me to get rid of?
Carcinogens.
If you get rid of mutagens, you get rid of all the carcinogens. Do you want to mutate? You do not. So this is better because it gets rid of all mutagens, which are all carcinogens or mutagens. So you get rid of chemicals that cause changes like this, which is not cancer.
Or some
Not extremely rare. I mean, you know, how much is the environment changing? You know, it's 1 every 250 million. It's not realistically healthy.
I thought you said earlier that not all mutagens are carcinogens.
That's right. Not all mutagens are carcinogens. This is not carcinogen. That's a mutation, not a carcinogen.
But all carcinogens are mutagens.
That's right. Anything that causes cancer is a mutation. And if you get rid of all mutagenic chemicals in your environment, you have stopped the change of your DNA and gotten rid of cancer. So the Ames test is incredible. And that is chapter 7.
So Dr. Hicks, so
Yes.
the Ames test will be a strain of bacteria that's lost its ability to synthesize histine?
Right.
Okay.
And that's what it uses, and it's a Salmonella oxytrope. Okay, so now we're going to talk about horizontally moving DNA, getting horizontal with DNA. And you're going to see a little video called gene transfer. And then we're gonna lecture over gene transfer. And then we're gonna see the recombinant DNA